Well, hi everyone. I'm Alan Schuldiner from the Regeneron Genetics Center and also the University of Maryland. And welcome to this second session, Genomics of Common Disorders, that I'm privileged to co-chair with my co-chair, George Patronos, who you'll hear from shortly. This is a really broad area for this second session, and we'll try to cover the landscape as best we can, from monogenic forms of common diseases, polygenic forms of common diseases, and also obviously the important environmental and lifestyle overlay of common diseases, genetically driven or not. And all of these things obviously vary quite a lot based on diversity in populations globally. So a really important topic to cover here in this G2MC virtual meeting. To kick off this session, we're going to have a keynote talk from Terry Manolio. Terry is from the National Human Genome Research Institute, and she's going to provide us an overview of a very interesting cohort consortium that has come together in the last couple of years, broaching these important questions of genetics of common diseases. She'll tell us about the International 100K Cohorts Consortium. Terry, please go ahead.
Great, thank you very much, Alan, and welcome everyone back from the break. I'm pleased to have the opportunity to talk about what we fondly refer to as the IHCC. The plan had been, because these two groups are so closely related, and actually G2MC is sort of a parent organization for IHCC, the plan had been for us all to meet together in Santiago, but of course plans change rapidly. So next slide, please. So this group was brought together based on the premise that there were a number of very large cohort studies that had been established and were being established in several regions worldwide. And each of them, large as they are, potentially several of them over 1 million participants, are still constrained by, to some degree, by their size, as well as to the ancestral origins and geographic boundaries in which they're operating. And these constraints can limit the subgroups or exposures or interactions that each of them can examine. So combining data, if that were possible, from all these cohorts would permit addressing pressing global questions that none of them can answer alone, and enhance their value, as well as leveraging the existing investments. Next, please. Okay, and so the vision then is to create a global network of large cohorts using multidimensional data from diverse populations for translational research that can be utilized to the maximum to enhance scientific understanding of a variety of contributors, not just genetic, but biological and environmental as well, to disease and population health. Next, please. Thanks. So this effort began about 5 years ago. The National Institutes of Health, one of the offices within the Office of the Director, compiled information on very large cohort programs defined somewhat arbitrarily as more than 100,000 participants. About 2 years later, the organization called the Heads of International Research Organizations, which is an informal coming together of groups that fund research internationally, includes some leadership from the NIH and Wellcome Trust and the Japanese AMED and a number of other groups, agreed to bring the cohorts together in an initial meeting, try to encourage data sharing, efficiencies, and maximize those investments. In October of 2017, the Secretariat of your Global Genomic Medicine Collaborative was invited to participate and lead this effort by identifying cohorts and organizing and hosting this initial summit. That first summit was held in March in the US. Second one was held in Reykjavik a year later, and the third one is now being, or just was held virtually in virtual Santiago. Next, please. So if you go to the Global Genomic Medicine Collaborative website, you'll see, click please, you'll see that IHCC, thank you, IHCC is part of that website. Click again, please. And if you click on that, you'll see a page come up that describes the IHCC and the various subgroups. And once more, please click. and then a list of the cohorts that have been identified. So there are a number of other resources here, but just to give you an idea of the scope and breadth of this effort. Next, please. And so the first summit was held. It had support from the Wellcome Trust, the British Medical Research Council, NIH, and the All of Us research program. And the goals really were to sort of explore how collaboration and compatibility might be maximized among these cohorts, discuss potentially putting together a searchable registry of global cohorts, and then understanding and perhaps addressing barriers to data and specimen sharing. Again, initially and somewhat arbitrarily, we set criteria for participation as having 100,000 or more participants. These participants were not selected for a disease, so this wouldn't be a cancer registry or something like that. It would need to have participants unselected. Biospecimens would need to be available or at least planned to be collected, and there needed to be the potential for longitudinal follow-up. Next, please. And it was recognized in that summit that there are compelling scientific questions that can be addressed with millions of individuals, such as the risks associated with rare exposures, such as running into a zebra outside of Africa, and the outcomes of the rare outcomes as well, generalizability of risk factors and associations, population-specific determinants of health, social and cultural determinants of health, potentially country or cohort-specific risk predictors, the impact of changing environments through migrant studies and other efforts, and potentially looking at inbred populations or other kinds of unique efforts to identify people carrying knockout mutations and non-functional genes for phenotyping of those individuals. Next, please. The overarching goals that were decided on in bringing the group together, who did agree to work together, were threefold: developing the information technology to promote sort of discoverability. So what is it, what cohorts are out there, and what information do they have? Develop a robust scientific plan that leverages cross-cohort collaboration and maximizes the input globally from the scientific community, including y'all. And developing the policy agenda to advance data sharing, engagement with industry, publications, etc. Next, please. We established 3 task teams, one led by Philip Awadala and Thomas Keene on data standards and interoperability that were charged with delivering IT standards and infrastructure to enable accessibility of data and cohort descriptions across international borders, a scientific strategy and cohort enhancement group led by Adam Butterworth, Hauken Haukerson, and Gadi Reinhardt, who's on this call. And the charge there was to develop a scientific agenda for novel approaches to diagnose and treat genomic conditions, as well as other conditions, explore enhancements to existing cohorts, and address diversity gaps. And then a policy and biodata sharing group led by Gunpegi Knudsen and Laura Rodriguez, charged to develop a policy agenda to facilitate and optimize impact of assembling the cohorts. And Laura, of course, is also leading the policy group for G2MC, so there's a good interaction there. Next, please. We held a second summit in Reykjavik last April. The number of cohorts grew from about 60 to 67. The number of countries increased, and the number of representatives participating, as well as 10 heads of, or international research organization groups also participated. Their acronyms are shown there. It's a little hard to follow, but at any rate, we did have robust interest from large numbers of international funders. We developed information technology and policy plans that would support the science, and there was strong interest in developing a formal collaborative structure and a charter. There had been several biopharma and sequencing tech firms, as well as other industry groups that had expressed interest in participating. We hadn't included them in the second meeting, and there was agreement that that was a good thing to do, and so they were, the group was then expanded to include them. Next, please. And then the third international summit was planned again for earlier this week. The idea was to coalesce around a visionary charter and a path forward, introduce a prototype, at least, of a cohorts atlas to enable collaboration among the cohorts, plan for developing a global toolbox of best practices to share among cohort studies, engage the full membership in charting a scientific agenda and identifying appropriate cross-cohort research projects, and then develop a scientific agenda to bring forward to funders. Next, please. But of course, in the meanwhile, next please, COVID-19 was making its way across the world. And so we as well converted to a virtual meeting. Next please. Which was held earlier this week, and we focused our objectives on galvanizing around the charter, but we added an objective of rapidly mobilizing cohorts to address the COVID-19 pandemic, and pivoted, I think, rather quickly to try to do that. We introduced the Cohorts Atlas and engaged really the entirety of the membership in developing scientific and policy agendas. Next, please. The charter is actually a quite mature document. Mary Da Silva, the Wellcome Trust, and John Connolly of the Children's Hospital of Philadelphia did an outstanding job in bringing this together. There was wide agreement that it should have guiding principles of transparency, fairness, equity, and inclusivity, much like the G2MC. It's a living document and designed to be guidelines rather than any kind of a legal contract. And it lays out requirements and expectations for collaboration, a governance structure, and various membership types. A publications policy was also proposed and largely agreed upon, along with policies for collaborating with industry and for data sharing. The data sharing principles largely based on the Global Alliance for Genomics and Health, which has been a partner from the beginning in the IHCC, and Peter Goodhand of the GA4GH is one of the three co-leads of the IHCC. Next, please. And just to very briefly describe a ton of work that went into developing a working prototype atlas that basically cataloged all of the participating IHCC members, including a lot of metadata about their cohorts, their establishments, the data that they collect, their research functions that were described and demonstrated that are robust and flexible, and a harmonization scheme that is also working quite well. The plan now is to further populate that with data from more of the IHCC cohorts and to establish a compelling set of research and clinical applications. There's my time, so I will go quickly. Harmonize the data dictionaries, add cohort phenotyping information, and pilot a polygenic risk score project. Next, please. And research topics that the group was quite interested in included, related to COVID-19, included the global expansion and timing of the epidemic, which can be addressed in international cohorts, the impact of the host genome and environmental exposures on manifestations of the disease, the impact of mitigation efforts on mental health and anxiety, and standards and methods for biospecimen collection. Next, please. And I'll stop there. And just many thanks to the people who made the IHCC possible and the 97 cohort leaders and participants that are sort of shown on this map. I'll stop there. Thank you.
Thank you so much, Terry. I think we have time for a question or two from the audience.
There is one question in the Q&A. Are you coordinating the efforts with other initiatives like ICDA?
Yes, so the ICDA actually gave two presentations at our IHCC meeting. The ICDA is the International Consortium or Coalition, no, something of Common Disease Alliance. International Common Disease Alliance, I believe. When they started up, we talked extensively about how we could integrate our efforts. We see ourselves as largely, a potential partner in identifying primarily genetic, but other determinants of health and disease that they will then use in trying to determine mechanisms, and then go from there. Their hope is to go from there into treatment. So their mantra is sort of maps to mechanisms to medicines. And I see how diverse is the collective population. I assume you mean across all of the IHCC cohorts. It's not as diverse as we would like it to be. Many of the cohorts are, and the largest ones are from the U.S. and the higher income countries. We have relaxed the criterion for number of participants for areas that are underrepresented, and so many of the participants, and you might have seen on the map that there were cohorts represented from Africa, South America, and other underrepresented areas. Most of those are much smaller than 100,000, but we are delighted to have them. And we actually heard about a couple earlier today that we would love to have join us.
So Terry, I attended the IHCC virtual meeting earlier this week. I was really impressed with the efforts towards data harmonization, particularly with regard to phenotypes. I was wondering if you might want to say a few more words about that process.
I would love to. I'm not in any ways knowledgeable about how one goes about doing this. I know it was a fairly labor-intensive effort. A lot of it needs to be done manually, which is a huge problem. Yeah. And it was largely led out of the Ontario Institute for Cancer Research, Philip Awadallah, and Thomas Keane at the European EMBL. Something laboratory, but at any rate, they have a lot of expertise in doing this kind of mapping across phenotypes, and are working in the GA4GH effort as well as in the IHCC effort. And I would encourage us to try to take advantage of the work that they've done, all of which is freely available, to see if we can't do something similar with G2C phenotypes.
Thanks so much, Terry.